Okay, this is lecture 6 in microbiology. We're going to talk about microbial growth. Microbial growth, when it comes to bacteria, bacterial organisms, refers not to cell size, but to the number of cells. So an increased microbial growth means an increase in number of actual bacterial cells. There are certain physical requirements needed for growth to occur, and they include within a certain temperature range, certain pH, and certain osmotic pressure. Two important terms that you need to understand and be familiar with are obligate and facultative. An obligate aerobe, for example, must grow in aerobic conditions. An obligate anaerobe has to be in anaerobic conditions. There's no freedom there of going back or forth, whereas an organism that is facultative can tolerate a particular condition. So obligate means that it's an absolute requirement, and facultative means that it can tolerate certain range, quite a wide range in those conditions. So anything obligate temperature. It has to be some organisms have to obligate high temperature. Okay, let's go into some more detail about temperature. There is typically an optimal temperature growth, and it's always a range. It's not one number exactly. So there's always an optimum growth temperature. There will be a minimum growth temperature below which the organism cannot grow, in other words, divide and increase in number, and a maximum growth temperature. And there are terms that are used for the preferred temperature range of organisms. Psychrophiles prefer colder temperatures, mesophiles medium temperatures, and thermophiles hot temperatures. And it's actually quite amazing just exactly how cold an organism can be and still grow, and how hot conditions can be, and yet the organism, if it's a thermophile, how hot it can be, and yet the organism can grow. Now I'm not restricting what I'm saying to organisms that grow in humans, that can infect and grow in or grow on humans. I'm talking about out in the environment. There are some real amazing extremes that prokaryotic organisms can thrive in. And here just gives you a brief idea. I wouldn't. Some sources would say that mesophiles actually is from 20 to 50. It varies. I mean, these are general terms, but certainly when you get into thermophiles and even hyperthermophiles, which does exist at in the mid-Atlantic, at the mid-Atlantic ridge at the bottom of the ocean, where the pressure is extremely high, the water temperature is above 100 degrees centigrade, and yet some organisms can still grow. In terms of growth of organisms and the ranges, the optimal growth range could be, and this would be an example of, I suppose, a mesophile in here. You get rapid growth bacteria. Some may, if they're pathogenic and growing in humans, may produce toxins that mediate the toxic effect. But again, I'm not restricting what I'm saying just to organisms that grow in humans. Some organisms, when you cool them, they'll survive and may even grow slightly. Others will, once they're in the temperature range of a refrigerator, and that's above zero, but well, the average usually is -4 plus or -2 degrees centigrade for a refrigerator temperature. That can slow growth tremendously, and so you get a slowing in the growth of spoilage bacteria that will spoil food. But there are very few pathogenic organisms that will grow in that temperature range. And so, for example, you may have milk, and milk is sold not sterile but pasteurized, and we'll talk about that later in the course. It undergoes pasteurization, but it is not sterile. It could have some bacteria in it. So you keep it in the fridge, which also slows the growth, but eventually you can get the milk spoiling because of bacteria that are slowly growing in there and can tolerate those temperatures. But it is extremely unlikely, especially after pasteurization, that you would have any pathogens in there, and very unlikely, even if they were in there, that they would grow because of it's at refrigerator temperature. Below freezing, there's never any significant growth, really, unless you're in extreme environments where it's naturally below freezing point of water. Temperatures in higher ranges will slow bacterial growth, and even higher will kill them, will destroy most bacteria. In terms of pH, the acidity or alkalinity of a environment, most bacteria grow actually typically between pH 6.5 and 7.5, although the fungi, if you look at fungi, which are not prokaryotes but eukaryotes, they actually will grow in more acidic conditions, typically pH 5 to 6. The term used for organisms that can grow in acidic, particularly in acidic environments, is acidophiles. In terms of the osmotic pressure, that refers to the tonicity of the surrounding medium that the organisms are in a hypertonic environment, where you have a high level of dissolved solutes in the surrounding solution. And that is not necessarily only salt. It could also be sugar. Any solute that's present, dissolved in the surrounding, well, typically it would be water, in the surrounding water solution, can have an effect on the osmotic pressure. And a hypertonic solution would be one that has high levels of solutes in it, higher than the normal inside the bacterial cells, and that would tend to cause osmosis, in other words, water to move out of the organism into the environment. And so that leads to a process called plasmolysis. And I'll show you a picture of what plasmolysis looks like on the next slide. Essentially, the cell membrane shrinks. The cell wall is very stiff. The bacterial cell wall is very stiff, so it retains its shape, but the cell membrane and its contents will shrink. That's plasmolysis. In the case of some organisms, they are able to survive and even grow in high tonicity solutions. If you look at, if you go to water bodies where it's only incoming water, there's no outlet, and the water is evaporating. The Great Salt Lake, Dead Sea, there's a number of areas like that in the world where you have really high salt concentrations. There are bacterial organisms, there are microorganisms that will grow in those conditions, and they are called halophiles. So they not only like but require a high osmotic pressure in the surrounding solution. That is, if they are obligate. If they are obligate halophiles, then they actually require a high osmotic pressure. If they're halophiles, that means they can tolerate high osmotic pressure. So facultative halophile could tolerate the high osmotic pressure. Obligate would absolutely need it. And here's a diagram showing the process of plasmolysis. This is a normal isotonic saline, 0.85% NaCl solution. It's a bacterial cell. I would say it's a gram-positive. How do I know? Because the diagram shows a single plasma membrane with a cell wall exterior to that. Remember that gram-negatives have an outer membrane as well. And here you see the cell membrane pulling away because of the movement by osmosis of water out of the bacterial cell into the surrounding high tonicity hypertonic solution. Are there other things that organisms need to grow? Yes, there are chemical requirements for growth, including all of these, and we'll talk a little bit about some of them. In terms of carbon, all organisms need carbon. Every living organism needs carbon to build up the organic molecules that it's made of, and they use one of two possible carbon sources. If they are able to take carbon from carbon dioxide in the air, then the organism is called an autotroph. If they need to take in carbon from organic molecules which were made by other living things, in other words, if they take them up from things that are dead, and they can take in the broken down products, then they are called heterotrophs. So organisms that use organic molecules for the carbon source are heterotrophs. Remember, there are two ways in terms of nutrition that organisms are classified. One is in terms of its carbon source, and the other is in terms of its energy source. And autotrophs use CO2 as a carbon source, and heterotrophs use organic molecules as a carbon source. And in terms of the energy, if they use light to generate energy, then they are phototrophs, and if they use organic molecules, then they are chemotrophs. So chemoheterotroph would use organic molecules as the energy source, and organic molecules as the carbon source. A chemoautotroph would use organic molecules for an energy source and CO2 as a carbon source, etc. We, of course, are chemoheterotrophs. Bacterial organisms could be chemoheterotrophs, or well, most of the ones that are important actually are, in fact, chemoheterotrophs. Important in terms of living in humans and causing disease in humans. They are all, in fact, chemoheterotrophs. There are important chemoautotrophs and photoautotrophs out there in the environment, but most of what the organisms all talk about are chemoheterotrophs, and we are chemoheterotrophs, of course. In terms of nitrogen, nitrogen is present in all amino acids. That's why it's called amino. Remember, an amino acid, which are the building blocks of protein, there are 20 different amino acids that go into making up proteins. The different proteins have different arrangements of the amino acids, different numbers of amino acids, and because of the side chains, they will fold into different shapes, and shape determines the function. Each amino acid has a central carbon atom with an amino group, a carboxy group, a hydrogen, and a R side chain. The R side chain is the one that differs between the 20 different amino acids. Anyhow, obviously, if there's an amino group in every amino acid, there's nitrogen, because amino refers to the presence of NH3 group on the side. Now, most bacteria will break down and decompose proteins, and some bacteria will use ammonium as a source for nitrogen, and others will use nitrate as a source for nitrogen. There are a few out there, not that grow in humans, but grow in a symbiotic relationship with plants that will use nitrogen gas. And so when when they talk about crops like alfalfa or clover or certain legumes, beans, that can put nitrogen actually into the soil, it's because in their roots there are bacteria that live which can pull nitrogen gas from air and pull it into the plant, into the organism, and that way into the plant, and use that as a source of nitrogen for growth. So they actually bring nitrogen into the soil. But for most organisms that are important that we'll discuss, they use either ammonium or nitrate for nitrogen source. There is sulfur in 2 amino acids. 2 of the amino acids contain sulfur groups in their side chains, cysteine and methionine, although it is also present in other molecules. And most bacteria, as I've said before, will decompose amino acids or proteins. And some bacteria use these amino acids as a source of their sulfur. Others use different sources, but they need sulfur, obviously, because they themselves have proteins to make, and therefore they need sulfur to make those amino acids containing sulfur. Phosphorus, of course, is important because it's part of nucleic acids, DNA, RNA, ATP. It's present in phospholipids of membranes also, and the source is phosphate, and organisms will need that. There are trace elements that are important, usually as enzyme cofactors. Things like magnesium, calcium are required as cofactors for active, in order to have activity of certain enzymes. The enzymes will not be active without them. So organisms need those. Now, in terms of oxygen, we can think of oxygen as either extremely important and absolutely required, or a poison. It varies, and there's a whole range in between, depending upon the nature of a particular species of bacteria. Remember, obligate refers to an absolute requirement. Facultative means that there's some tolerance and range. So an obligate aerobe, and these are examples of what I've talked to you about before, which are stab cultures. If this is a test tube filled with nutrient agar, and a person in the lab takes a sample of a bacterial culture at the end of instead of a loop, a needle, a long inoculating needle. So the sample is taken at the tip of that, and then they stab it into this culture to inoculate the culture. Well, then you'll get growth all the way along, or just at the bottom, or just at the top. It varies depending upon the response and requirements for oxygen for growth. And here you see the result of inoculating down. This is called a stab culture, and all the examples down below are, in fact, examples of stab culture technique. What we see here are an obligate aerobe growing. So it only grows towards the surface where there is some oxygen that can penetrate down into the agar. A facultative anaerobe, the organism's it grows at the surface, but it's facultatively anaerobic, so it can also grow further down into the organism. An obligate anaerobe would only grow at the bottom. I'm not going to talk about aerotolerant anaerobes. It's a bit of a argument between aerotolerant anaerobe and a facultative anaerobe. And the term's actually not used that often. Microaerophiles, they have a requirement for oxygen, but it's at a level that is below atmospheric levels of oxygen, and so they'll grow at a mid-level. They need some oxygen, so they won't grow at the bottom, but higher levels of oxygen are in fact not tolerated by, they're unable to grow at normal atmospheric levels. And a good example of an organism that is a microaerophile would be species in the genus Helicobacter. Helicobacter is an organism that was first described in the 1980s. Helicobacter. This is the genus name. There are many species members of this genus, and the important one for you guys to know about, and we'll talk about it later into the course, is Helicobacter pylori, which can cause pyloric ulcers and even lead to cancer in the stomach. Okay, so I told you that in fact oxygen can be toxic, and not only oxygen but molecules that contain oxygen. There are certain types of molecules that contain oxygen that are highly toxic, and they're even in fact used by cells in our body to kill microorganisms. The toxic types of oxygen containing molecules that phagocytic cells produce, and this includes things like superoxide, which is shown here, superoxide, peroxides, and hydroxyl radical. And superoxide is actually the full name. It should say the full name here. I'm sorry, I left it out. Superoxide anion. A lot of books will say just superoxide, but the correct name is superoxide anion. Superoxide anion, and the chemical formula is O2 minus. Peroxides are O2 and 2 minuses, although no, O2 2 minus. And hydroxyl anion is typically shown like this. It's a hydroxyl group with a free electron, shown as a single dot. A free electron is associated with an unpaired electron associated with the group. Let's not get into that too much. What you should know is that, in fact, phagocytic cells in the body, which are the main cells that protect us in terms of the non-specific immune response, and the very first cells involved in the inflammatory response, are able to produce these toxic oxygen, toxic forms of oxygen, and thus kill the microorganisms that they take in or phagocytose. The molecules, in fact, these superoxide, peroxides, and hydroxyl radical that can be produced by macrophages or neutrophils are in fact so toxic that they will kill the cell itself. Not only the organism it phagocytoses, but also the cell, the phagocytic cell, the macrophage or neutrophil, is in fact itself killed by these products. Now that may seem self-defeating, but it's not. In fact, what it does is it's a mechanism that the body has to limit this kind of very strong inflammatory type of response. It is a response that's required in order to defend against any microorganism that gets into the body. But at the same time, it is in fact so intense and requires production of such toxic molecules that the toxic molecules will kill the cells that move in, and thereby limit the inflammatory response. If the organisms are not all killed and are still growing, and by growing, when I talk about microorganisms, I'm talking about increase in cell number. Bacteria will increase in cell number when they grow. Then new phagocytic cells will move from the bloodstream into the area where the bacteria are present in the body, and they will be activated and produce these toxic oxygen molecules. So what you get is a progressive accumulation of cells that phagocytose and kill themselves, And if they've done a complete job and killed off all the bacteria, then there's no more signal for new cells to move to the area. The cells that accumulate and die are dead white cells, and it's called pus. So that's a very nice mechanism that the body can use to limit, to have a very intense, strong response and kill organisms, and yet at the same time limit the inflammatory response so that it doesn't carry on and injure surrounding tissues. Kill the bacteria, but stops once that has been accomplished. Okay, there's a little bit more information about these molecules that I'd like you to know. One is, well, the fact that on the previous slide, they're all produced by phagocytic cells, and will kill bacteria, but can also kill the cells themselves that are producing it and therefore limit inflammation. But also that, cells also, bacterial cells have some, depends which one you look at, have evolved mechanisms to detoxify these kind of defense molecules. So superoxide anion, for example, some organisms have the enzyme superoxide dismutase, and superoxide dismutase will cause superoxide anion to be converted to hydrogen peroxide. Well, you may say to yourself, well, hydrogen peroxide is also toxic, but such cells may also contain an enzyme called catalase, and catalase converts the hydrogen peroxide to water and oxygen. So one of the tests that we carry out in the lab, we won't do this one in the lab, we don't ordinarily do it, but one test is to look for catalase activity in an organism in colonies growing on a plate, because it shows whether they can detoxify hydrogen peroxide, and it also helps with the identification of the organism, because not all organisms, not all bacteria do produce catalase. Another way that some organisms detoxify hydrogen peroxide is by producing the enzyme peroxidase, which will again form water in these reactions. You don't need to memorize the reaction, but know, please, that detoxification of superoxide can occur with superoxide dismutase, and then peroxides like hydrogen peroxide can be detoxified by catalase or peroxidase enzymes. Hydroxyl radical, another highly toxic oxygen containing molecule, the most toxic of all of these mentioned, these three. I don't know that there is anything that will detoxify it that's produced by a bacteria. They are all susceptible to the toxic effects. It's extremely toxic. All of these molecules, superoxide, the peroxides, hydroxyl radical, there's a general term that's often applied to them that I didn't put on this slide, but they're often called, oh dear. Oh yes, they're often called oxygen radicals. Okay. A radical is an extremely reactive kind of molecule, and an oxygen radical is just an extremely reactive molecule that contains, where the reactive group is the oxygen. So oxygen radicals. All of these are oxygen radicals. Very toxic and quite effective at killing microorganisms, and also, interestingly enough, produced by phagocytic cells of our nonspecific immune system, used again to kill the bacteria or microorganisms that they phagocytose. There are in another kind of other requirements for growth of bacterial organisms, prokaryotic organisms, or microorganisms. There are many growth factors that are required. Enough said about that. Okay, I think that this would be a good place to stop lecture 6, part 1.